Thank you all the participants. So in the last session, we have covered a few basic concepts and definitions of genetic toxicology, then types of mutations, importance of genetic toxicology assessment, the case study, and we have covered two important assays. First is a bacterial reverse mutation assay, and second was in vitro chromosomal aberration assay. So I will give a quick overview what we have covered in the last session. What is gene, chromosome, and the DNA? So, in a simple language, DNA is a long piece of string. In those string, there are knots. So, those knots are the genes, and a big coil of the knots are chromosomes. So, chromosome is a thread like structure which is located inside the nucleus of animal or plant cells. And each chromosome is made up of protein and a single molecule that is a DNA. So, genetic toxicology is a study of how chemicals, radiation, or the environmental factors that can cause a genetic damage in the living organisms. We have also covered the key concepts what is mutation, mutagen, mutagenic, mutagenicity, then genotoxicity, difference between the both genotoxicity and mutagenicity, also carcinogenicity, difference between carcinogen and the mutagen, then clastogen, that which cause the structural chromosomal aberrations, then anugens, which cause a chromosomal misaggregation leading to aneuploidy, that is abnormal chromosome number in cells. We have also covered the somatic and germ cell mutations. Then we have covered the types of mutations based on the cell type, that is somatic and germ cell, difference between the two. So somatic causes the local effect, and germline causes entire organism will be affected because of the germline mutation. Then mutations based on the structural changes, point mutation, frame shift mutation, then chromosomal mutations. Then functional consequences of the mutation, that is silent mutation, missense mutation, and nonsense mutation. Then mutations based on the origin, that is spontaneous and induced mutation. We have also covered one case study for the groundwater metabolites. And then the next part was the bacterial reverse mutation assay. So I got some recommendation to include some genotypic characterization of the bacterial reverse mutation assay. So in the bacterial reverse mutation assay, the objective is to detect the mutagenic potential of the test atom. For that purpose, as per OECD 471, it is recommended to include all 5 tester strains. So first is the Salmonella typhimurium, TA97A or TA97 or TA1537 you can include. The next is the TA98, TA100, then TA1535, or you can include TA102 or E. coli WP2. So why these strains are important? Because each strain have different characteristic. So you can see there is a base pair substitution in TA1535, TA100 and E. coli. There is a occurring mutation in TA102, and there is a frame shift mutation in TA1537, TA97A and TA98. So all these strains have a defective enough DNA repair capacity except TA102 or an E. coli WP2. This is the first characteristic. Then the second is all these tester strains have a deficient lipopolysaccharide barrier on the cell wall except the E. coli WP2. So these two properties gives the extra sensitivity to DNA damage, and it also increases the cell permeability. So large molecule can easily penetrate uh, into it. Then the next is a testosterone TA98, T100, TA102, TA97A. It contain a resistant transfer factor, that is plasmid PKM101. So this factor confers resistance to ampicillin, so which enhances the operation of error-prone repair system. Then in case of TA102, so it contain a plasmid PAQ1, so which confers the resistance to tetracycline. For testosterone TA1535 and E. coli, uh, 1535 TA100 and E. coli. So these are highly sensitive for the base pair substitution, as I said earlier. And TA1537, TA97A, TA98, those are sensitive to frame shift mutation. Then TA102, which contains a OCRA mutation, that is a nonsense mutation. There is a addition of the stop codon there. So these are basic genotypic characteristics of testosterone used in the bacterial reverse mutation assay. I have already covered in vitro chromosomal aberration test. Its objective is to evaluate the classogenic potential of the test item, and OECD test guideline OECD 473. So today I am going to cover next part, that is in vivo chromosomal aberration test. So objective of this test is to evaluate again the classogenic potential of the test item, means structural chromosomal aberrations. For that purpose, you can use a test system rat or mice, and test guideline is OECD 475. So to conduct this assay, it is necessary to include the concurrent vehicle as well as positive controls. So what is the study flow? So first part of any assay is the solubility of the test atom, which is a most crucial part. So for in vivo chromosomal aberration test, you can use different vehicles, range of vehicles. So first choice of vehicle is of course aqueous vehicles. So you can use analytical grade water or analytical grade water with 0.2% vinity. If both are not suitable for the test item, then you can use oil. For example, corn oil you can use. Then the next part is lethal dose 50. So you can check the reported literature or published data for the LD50 value. If LD50 is greater than 2000 mg per kg body weight, then there is no need to conduct the dose range finding study. If LD50 is less than 2000 mg per kg body weight, then you have to conduct the dose range finding study with multiple doses with both male and female mice. So first part is a dose range finding study or the exploratory study. If LD50 is greater than 2000 mg per kg body weight, so after dose range finding study, you get based on the clinical science and the mortality and percent cytotoxicity, you get the value for the main study or doses for the main study. So for the main study, it is necessary to include the concurrent vehicle control, which is suitable for this for the test item. Then at least three test concentration if dose is not limit dose, and the positive control, maybe cyclophosphamide, I can consider known clastogen. And during the after the conduct of the main study, it is necessary to evaluate the slides using microscopic method for the in vivo chromosomal aberration. In vivo chromosomal aberration, there are two sampling methods. First sampling, there is a vehicle control, positive control, and one dose for limit study. And if it's a multi-dose study, then vehicle positive and at least three dose levels. And there is a second sampling of vehicle and only one dose. So why bone marrow cells are selected? So bone marrow cells, particularly from the femur, those are rich in the rapidly dividing cells. So it makes them ideal for detecting the chromosomal aberrations after the exposure of the test item. Here we have included two sampling methods. So there is a significance of two sampling times here. So two sampling times ensures chromosomal damage is detected across different stages of the cell division. So it gives the variability in how quickly different animal can metabolize and can respond to the test item. So there are different reasons why two sampling times are selected. First is a timing of the cell cycle. So chromosomal aberrations are visible during the metaphases. So since these cells all the cells don't all divide at the same time. So if there are two sampling points, it increases the chance of catching the cells at right stage. So two sampling points are important. Then second is a metabolic differences. So few substances those act very quickly and they get metabolized fast than the others. So two time points help to capture both early and delayed effects. So metabolic differences we can catch here. And the third is a regulatory guideline. So third point is a regulatory guideline. So regulatory guideline OECD 475 recommends the multiple sampling. For example, 12 to 24 hours post treatment to improve the reliability and sensitivity of the test. So this is the second reason of two sampling times. So in this diagram, you can see chromosome damage in the bone marrow of mouse. So you can see the chromosome break here. Maybe picture is not clear. Then chromatid break is here. Then there is a dicentric is here. Then there are fragments, acentric fragments. Also there is a ring chromosome. So the next assay is a in vivo micronucleus test. So again, the objective of in vivo macronucleus test is to evaluate the clastogenic potential of the test item using mice peripheral blood or the bone marrow. So you can use a test system rat or mouse, and the test guideline is OECD 474. So again, in this assay, it is necessary to include a vehicle and concurrent positive controls during the experiment. Then study flow is again same, that is row stage finding study or exploratory study, then main study and slide evaluation or the flow cytometric analysis. So before going into the detail, we should understand what is micronuclei, what is mature erythrocyte, what is immature erythrocyte, and what is erythroblast. So micronuclei are the small nuclei present those are the small nuclei basically, and those are separate or additional from the main nuclei of the cells, and those are produced during the telophase of mitosis by the lagging chromosome fragments or the whole chromosomes. Then the next part is a normochromatic or mature erythrocytes or NCEs. So NCEs are fully matured erythrocyte, so that has lost the residual RNA and which remains after enucleation or lost other short-lived cell markers. Those are characterized disappear after enucleation following the final erythroblast division. So normochromatic erythrocytes or NCE in a simple language, those are the matured erythrocytes, and in opposite polychromatic erythrocytes, those are immature erythrocytes or those are the newly formed erythrocytes. So those are formed in the intermediate stage of development and which stains with both blue and red components of classical blood stains. Then the third part is the erythroblast. So erythroblast means an early stage of erythrocyte development immediately preceding the immature erythrocytes where the cell still contains a nucleus. So how this micronucleus form? So micronucleus form when the chromosome or the chromosomal fragments fail to be included in the main daughter nuclei during the cell division. So this happen typically due to the two main mechanisms. First is a clastogenic event and the second is an anogenic event. So, what is clastogenic event? So, in the clastogenic event, it involves the physical break of chromosomes, often caused by the DNA damage due to radiation or the chemical or the oxidative stress, and it results in the formation of the acentric fragment or the fragment which lacks the centromere basically. So it due to the lack of the centromere, it cannot attach to the spindle, and it left behind during the anaphase, and it eventually formed the micronuclei. So this is first a clastogenic event. Then the second reason or mechanism is anogenic event. So in anogenic event, it involves a whole chromosome that lag behind due to errors in the matritic spindle or kinetochore malfunctions or problem with the chromosome segregation. So, in the anogenic events, these are lagging chromosomes also get enveloped separately, and it forms the micronuclei. So, which is separate from the main nucleus. So, this is a diagram of chromosome formation, micronucleus formation. So, micronucleus formation due to chromosome malsegregation or unrepaired breaks. So, a nucleoplasmic breach or micronucleus formation due to DNA strand break, misrepair, incomplete chromatid separation, or telomere end fusion. So, a whole chromosome or some part of the chromosome which lag behind, and it forms a micronucleus. So what is the purpose of the micronucleus test? So purpose of this test is to identify the substances that can cause cytogenetic damage, which result in the formation of micronuclei containing entire either lagging chromosome fragments or the whole chromosomes. So when a bone marrow erythroblast develop into a immature erythrocyte, which we referred as a PCE or polychromatic erythrocyte, the main nucleus is extruded, and any nucleus that has been formed may remain behind the cytoplasm. So, visualization or the detection of that micronuclei is facilitated in these cells because they lack a main nucleus. So, if there is an increase in the micronucleated immature erythrocytes, that is MNPCEs, in the treated animals compared to vehicle control, it is an indication of induced structural or numerical chromosomal aberrations. So, in this diagram, you can see the PC is blue in color, the MNPCE, the small round inside the PCE, and the NCs are also there so that you can differentiate easily with the microscopic examination PCE, NCE, and MNPCE. So why focus is only on the micronucleated PCs rather than micronucleated NCs? So there are a few key reasons for that. First reason is PCs are newly formed cells. So PCs are immature red blood cells that have just exited from the bone marrow. And if they contain a micronuclei, it means that the chromosomal damage occurred recently. So it makes them ideal for detecting the genotoxic effects of a test substance. In contrast, NCs reflect the older events because NCs are more mature than the PCs, and they have may have been circulating for a while, and micronuclei in the NCEs are result from earlier or unrelated exposures, and so it make them more reliable for pinpointing recent genotoxicity. For those reasons, MNPCs are preferred over the MNNCs. Then the clearer signal. Since PCs are more sensitive indicators of recent chromosomal damage, evaluating MNPCs gives a more accurate and timely readout of the test compound's effect. And the last reason is regulatory guidance. So, standard protocol like OECD Test Guideline 474 recommended scoring MNPCs as the primary endpoint of these reasons. So I'm going to give the continuous flow of the study of the in vivo micronucleus test. So first part is a dose range finding study or exploratory study. Same like in vivo chromosomal aberration test. First part is the solubility of the test atom. First aqueous vehicles, analytical grade water or water with twininity or the corn oil. Then dose range finding study or exploratory study based on the LD50 value of the test atom or the clinical science and the mortality results. Then based on the cytotoxicity or a reduction in PCs over total erythrocytes. If a reduction is less than 50%, you can select that concentration for the main study. And for the main study, you should include but first a vehicle a concentration, maybe five animals, three to five animals. Then test concentration, minimum three test concentrations, and the positive control. And if there is no sex wise difference, then you can include only one sex for the evaluation. And there is a slide evaluation for the manual method for bone marrow basically. And if flow cytometric method, then it is automated method. So few recommended protocol for in vivo micronucleus test. So on day one, there is a treatment. Then again day two, the treatment with the clinical science like day one. Then on day three, again the clinical science, and on day four, the removal of the bone marrow, then staining and slide preparation, or the flow cytometric method, that is removal of peripheral blood, fixation, and incubation. So this is the general flow of in vivo micronucleus test. The next part is a in vitro micronucleus test. So instead of in vivo micronucleus test, you can conduct in vitro micronucleus test, which is a second battery assay after the AIMS test, mostly for the pharma compounds. So objective of this assay is to evaluate the clastogenic and anogenic potential of the test atom. So test systems preferred are human peripheral blood lymphocytes, or you can use rodent cell lines like CHO or V79, or you can also use a human cell line like TK6, human lymphoblastoid cell line. And test guidelines are OECD 487 and ICH S2R1. So for this assay, you should include concurrent vehicle control and positive controls. So positive controls should be known clastogen or anogens. So what is the principle of this test? So the cell cultures of human or other mammalian origin, those are exposed to the test system with and without exogenous metabolic activation system, that is with S9 and without S9. During or after the exposure of the test item, the cells are grown for a period sufficient to allow chromosome damage or other effects on a cell cycle or cell division to lead the formation of micronuclei in interface cells. Then the cells are harvested, those are stained and analyzed for the presence of the micronuclei. Ideally, the micronuclei should only be scored in those cells that have completed the mitosis during the exposure to the test item or during the post treatment period if one is used. If cultures in cultures that have been treated with cytokinesis blocker, this is easily achieved by scoring only binucleated cells. In the absence of cytokinesis blocker, it is important to demonstrate that the cells analyzed are likely to have undergone cell division based on an increase in cell population during or after exposure to the test chemical. So, first part of the in vitro micronucleus test again is a solubility test. So as per OECD 487, it is recommended that the highest test concentration should be 2 mg per ml or 2000 microgram per plate. As per ICH S2R1, the highest test concentration recommended is 0.5 mg per ml or 500 microgram per ml. 0.5 mg per ml or 500 microgram. So first part is the solubility. So first preferred vehicle is analytical grade water. If compound form uniform suspension in analytical grade water, then you should go for the other organic solvents. So first preferred solvent is dimethyl sulfoxide. Then second is ethanol or the acetone. You should achieve maximum solubility in the vehicle. After solubility test, the second part is a precipitation and the pH test. So you have to prepare the range of concentrations here. For example, you have to conduct a precipitation test. First, you have conducted the solubility test and the compound is soluble at 200 mg per ml. So now you are going to test five different concentrations ranging from 2000 microgram, that is 2 mg, 1000 microgram per plate, 500, 250, and 125. So you are going to add the test concentration in the plain media, and you should check the precipitation. If there is a heavy precipitation, then that dose should be rejected. If there is a slight precipitation or there is a no precipitation, you can include that dose for the further analysis. For example, if there is a heavy precipitation at 2000 microgram per plate, and if there is a slight precipitation at 1000 at 0 hour and after the 3 hours of incubation, and if there are no precipitation starting from 500 to 125, then for the study you can include 1000 microgram per ml as a highest test to conduct the PCYT study. So after the precipitation, it is necessary to conduct the pH test. So there should not be there should the pH of the test should be compared with that of the vehicle control. If pH is less or greater of or pH of the test solution is acidic or basic, you should adjust the pH with the help of buffering solutions. So after precipitation and pH test, you can select a dose for the cytotoxicity test. The next part is for in vitro macronucleus test. If we consider TK6 cell line uh, as a test system, then for the cytotoxicity test, first part is a target cell preparation of the TK6 cell line. So before conducting your study, your test system should be well characterized by the external agency, as well as in-house characterization is necessary. For example, chromosome number analysis, then doubling time, then mycoplasma contamination analysis. So it should not contain a mycoplasma, or it should be free from all the bacteria or fungal contaminations. Then chromosome number should be within a range, and doubling time should be achieved. So you should test all these parameters. So if the test system is well characterized, then only you can use that test system for the further analysis. So first part for the preliminary cytotoxicity test is a target cell preparation. After target cell preparation, you can prepare a range of concentrations for the study, and you can add target cells in the flask, and the test concentrations along with the vehicle control, corresponding vehicle control, the different flask, and then incubate for 3 hours, 3 to 4 hours, for without metabolic activation system, 3 hours exposure, and with metabolic activation system, 3 hours. Before break, I have covered the solubility test, precipitation, and pH test, and the next part was cytotoxicity test. Before that, I have covered the characterization of the test system. So test system should be well characterized before performing any assay. So in if you consider a test system that is a TK6 cell line, so first part is a target cell preparation. Then you have so there are three experiments. First experiment is without metabolic activation system for 3 hours exposure. Then second is without a metabolic activation system, 24 hours exposure of the test item. And third is a with metabolic activation system, metabolic activation system, 3 hours of exposure. So On first day target cell preparation, then treatment with the test range of test concentrations, and on the second day there is a cell count. So based on the cell count you can calculate the percent cytotoxicity. If percent cytotoxicity is between 50 to 60%, then you can select that concentration for the main study. For example, if highest concentration is 1000 microgram per ml and a percent cytotoxicity is about 80%. For 500 microgram per ml, if percent cytotoxicity is 60%, then for 250, if it's 52%, and for 125, if it's 40%, then for the main study, you can select highest test concentration, that is 250 microgram per ml, based on the results of 1000 and 500 as well. And you can test lower concentrations as well for the main study, that is highest concentration is 250, then 125, then 62.5 and 31.25 microgram per ml. If highest concentration selected for cytotoxicity is 2000 microgram itself, and if you do not get any cytotoxicity there, that is cytotoxicity is less than 50%, then for the main study you can directly include 2000 microgram per ml as a highest test concentrations. And in the main study, it is necessary to include vehicle control as well as known clastogen as well as mutagen. For example, you can use clastogen, mitomycin C, or cyclophosphamide monohydrate, and you can use anogen like vinblastine for the analysis. So the same methodology is there for the main study. Only after cytotoxicity, you have to evaluate the macronucleus formation uh, for human peripheral blood lymphocytes as well, and for other cell lines as well. The protocol is slightly different for human peripheral blood lymphocytes. For blood lymphocytes, first part you have to remove the blood from the human volunteers. So first crucial part is ethics committee approval for use of human blood. So you need approval from the ethics committee. Then you need a audio visual recording or written consent from those volunteers. Then you have to remove the blood from the volunteers, or you can directly procure blood from the external agencies. And then you can conduct hematology and clinical chemistry parameters, and you can submit those reports to the doctor to see whether the parameters are within a range, and you can use those volunteers for the study or not. And after that
Excuse
you
me.
can actually
Uh,
conduct the main study. Yes.
Slides are not moving.
Now.
Uh, no, those are old ones.
Okay. So for the human peripheral blood lymphocytes, the flow chart is not included in the presentation. I am only explaining it. Then you can basically culture the cells in the RPMI with antibiotic, and you can use PHA for cell division, and the rest procedure is almost same for human peripheral blood lymphocytes. The cytotoxicity parameter is same, that is 50 to 60% cytotoxicity should be there to select the concentration for the main study. So this is a diagram of micronucleus formation. You can see these are the binucleated cells, and there is a micronucleus inside. So in the in vitro micronucleus test, the binucleated cells are specifically scored because they provide a clear evidence that the cell has completed one mitotic division and only one. So this is crucial for accurately detecting macronuclei that result from chromosomal damage during that cell division. So binucleated cells are the gold standard for this assay. So first reason is cytokinesis block strategy. So a compound called cytokalism B is added to the culture to inhibit the cytokinesis. So we use cytokalism B when we are using a test system human peripheral blood lymphocytes. So it inhibits the cytokinesis. The cytokinesis means a final step of cell division where the cytoplasm splits without affecting the nuclear division, which results in cells with two nuclei, proof that mitosis occurred. And the second reason is a timing matters. By scoring only, there is a background noise. Thank you. So second part is a timing matters. So by scoring only binucleated cells, we ensure that any micronuclei observed form during the most recent mitotic event, not form earlier or multiple divisions. The third reason is it reduces the variability. So it standardizes the population being analyzed, which makes them more reliable and reproducible. And the fourth reason is OECD test guideline 487 recommendation. So 487 recommends this approach for precisely the reasons because it enhances the sensitivity and specificity of the assay. For that reason, we have only included a binucleated cells for the evaluation. The next part is a cell gene mutation assay. So objective of cell gene mutation assay is to detect the mutagenic potential of the test item. So you can conduct cell gene mutation assay using OECD 476, CHO-K1 cell line, HPRT gene. Else you can conduct using a OECD 490, that is mouse lymphoma cells. So you should include a concurrent again vehicle and positive controls for the assays. So those should be positive controls should be a known mutagens, like you can use benzopyrene or you can use mitomycin C or any other recommended by the OECD guideline. Then study flow is a first solubility, then precipitation and pH test. Like in vitro micronucleus assay, it is necessary to conduct first solubility test, and a preferred vehicle should be the plain media or the analytical grade water, that is aqueous media. If compound form suspension or not soluble in the aqueous solutions, then you can include other organic solvents for the assay like a DMSO or the ethanol or acetone, as per OECD guideline. And the highest test concentrations are same like in vitro micronucleus test, that is 2 mg per ml for OECD 476 or OECD 490. The next part is a precipitation. So same like in vitro micronucleus test, you should exclude the heavy precipitation doses, because what happened during the heavy precipitation, there won't be a proper exposure of the test item to the test system, so that you can get equivocal results there, or there is an interference of the test item. For the precipitation purpose, you should include the slight precipitation or no precipitation dose. And the next part again, the pH test. So for the pH test, if there is a variations in the pH, or if the pH is acidic or it's basic, you should adjust the pH using buffering solution. You can use a sodium bicarbonate buffer for that purpose. Then the next part is the cytotoxicity test. But before commencing the cytotoxicity test, again, we are going to use a CHOK1 cell line, and we are going to use a mouse lymphoma cells. So these cells should be well characterized. So you can procure those cells from known sources like ATCC or ECSCC. And so those ATCC or ECCC provide the characterization certificate of those cells, and after the receipt you can prepare the frozen permanents in house. So before the assay you should in house characterize the test system with the chromosome number, or the mycoplasma contamination is present or not, or cell leveling time is within a range or not. So you should do the in-house characterization of those test systems, and then you can conduct a cytotoxicity test. For both CHK1 and mouse lymphoma, the protocol is almost same. So first is a target cell preparation, and then you have to expose those target cell with a range of concentration during a cytotoxicity test. So there are again three experiments with metabolic activation system, 3 hours, 24 hours exposure, 3 hours exposure, and 24 hours exposure. So without metabolic activation system, 3 hours exposure, 24 hours exposure, and with metabolic activation system, only 3 hours of exposure. So after 3 hours of exposure, you should change the media, and for 24 hours exposure you should continue the exposure till 24 hours, and then after 24 hours you can directly seed the cells. After seeding on day 12 or day 14, you should count the colonies and compare those colonies with the vehicle control colonies, and then calculate the cytotoxicity. So if a relative survival is within 10 to 20%, then you can take that concentration, or you can select that concentration for the main study. Uh, for central mutation assay, if a relative total growth is between 10 to 20%, you can select that concentration for the main study. For example, if there is a cytotoxicity that of a percent relative survival is greater than 30%, for 2000 microgram per ml, you can directly select that concentration for the main study. If relative survival is, for example, 5% for 2000 microgram per plate, for 1000 microgram per plate, relative survival is 7%, and for 500 microgram per plate, if relative survival is 15%, then the highest concentration selected for main study is 500 microgram per plate per ml. For the main study, again, minus without and 24 hours exposure, 3 hours with and without metabolic activation, and 24 hour without metabolic activation, and you should include the vehicle control and positive controls. So this is a difference between the CHOK1 cell line and the mouse lymphoma cell line assays. So for the CHOK1 cell line, the target gene is HPRT gene, that is hypoxanthine guanine phosphoribosyl transferase, and for mouse lymphoma, it is a thymidine kinase gene. The OECD test guidelines are 476, and for mouse lymphoma, it is 490. So mutation type detected for CHOK1, the point mutations, base pair substitutions, and small deletions. And for mouse lymphoma, point mutations and large deletions. The selection agent is a 6TG for CHOK1, and for mouse lymphoma, trifluorothymidine, that is TFT. Then cell type, CHOK1 is a adherent epithelial like cell line, and mouse lymphoma is a suspension cell line. It's a suspension lymphoblastoid cell line. So metabolic activation system with and without metabolic activation for both CHOK1 and mouse lymphoma we should include. So sensitivity to antigen. So CHOK1 is a less sensitive to antigens, and mouse lymphoma is a moderate sensitive to antigens. Then a spontaneous mutation rate. So CHOK1 spontaneous mutation rate is low, and for mouse lymphoma it is higher than the CHOK1. Then application. So CHOK1 is used for general mutagenicity screening, and mouse lymphoma is used for broader detection of mutagenic mechanisms. What is the principle? So mutant cells deficient in thymidine kinase enzyme activity because of mutation, that is TK plus minus to TK minus are resistant to cytostatic effects of pyrimidine analog, that is TFT. So the proficient cells are sensitive to TFT, which causes the inhibition of cellular metabolism, which halts the cell division. and mutant cells are able to proliferate in presence of TFT and form a visible colonies, whereas that cells contain TK enzyme, they cannot. So in general, the cells in suspension are exposed to for analyzable test concentration of the test item, both with and without metabolic activation for a suitable period of time. Then they are subcultured to determine the cytotoxicity to allow phenotypic expression prior to mutant selection. So on day 0, there is a target cell preparation and there is a treatment with the test item. So on day 2, uh, on day 0 itself, after 3 hours of exposure, you can change the media. Then for 24 hours exposure, after the completion of 24 hours, you can change the media, and then you can seed the cells for the cytotoxicity determination. Then cytotoxicity is usually determined by measuring the relative cloning efficiency or relative total growth of the cultures after treatment period. The treated cultures are maintained in a growth medium for a sufficient period of time to allow near optimal phenotypic expression of induced mutations. Mutant frequency is determined by seeding known number of cells in a medium containing selective agents to detect mutant cells, and in medium without selective agents to determine cloning efficiency. After suitable incubation time, colonies are counted. So if we consider only 3 hours of exposure with and without metabolic activation system, so after treatment you can change the media on day 0. On day 1, again you take a cell count, and then again you change the media, take cell count, again passage the cells, and on day 2, on day 2, that is 48 hours after the treatment, you can again seed the cells for the cytotoxicity determination, as well as the mutant frequency determination. So for the mutant frequency determination, you seed the cells with known number of cells containing a selective agents, so that we can detect a mutant cells. And for the cytotoxicity determination, you can seed the cells without selective agent, that is TFT, for the mouse lymphoma cells, and 6TG for CHOK1 cells. So after the exposure, the colonies are counted, and colonies are compared again with the vehicle control and again with the positive control. So there is a specific criteria which is given by the OECD test guidelines. If those are statistically significant, results are statistically significant, then compound is mutagenic. If not, then compound is non-mutagenic. In this way, you can conduct cell gene mutation assay. So this is a diagram of large and small colonies in the cell gene mutation assay. You can see in diagram C, the more than 75% part of the well is covered. Uh, so this is a large colony, and this is a small colony, because less than 25% part is covered by the cell. Then the next part is the in silico approach, that is QSAR, or quantitative structure activity relationship. So for mutagenicity assessment, you can also use a quantitative structure active relationship. So there is a Vega software which predicts Ames mutagenicity using a consensus QSAR model, which combines multiple algorithms to assess whether a chemical is likely to cause mutations like in bacteria like Salmonella typhimurium. So how it detect or how it gives the structural alert. So you have to provide the chemical structure to the software. So in the software, it contains a library of compounds or structures, and it compare the structure with the known library. So model execution is there. So Vega runs the compound through submodels, and there is a consensus prediction. So it integrates the output to give a final call, that is mutagenic or non-mutagenic. So basically, it compare the structure with the library structures as well. So what is the applicability of the domain? So it checks whether your compound falls within the reliable prediction space space of the model or not. And structural alerts. So Vega highlights specific substructures like nitroso group or aromatic amines known to be associated with the mutagenicity. So these alerts are based on expert curated rules and statistical patterns from training data. So if you get a structural alert in the in silico approach or using a QSAR model, then you can directly conduct a first battery assay, that is AIMS test. So this is a in silico method for genotoxicity assessment. So we have covered few battery assays of the genotoxicity, that is AIMS test. Then second battery assay is in vitro chromosomal aberration test or in vitro micronucleus test. Then third is a in vivo micronucleus test or in vivo chromosomal aberration test. And fourth is a search gene mutation assay. And the last part is the in silico approach. So DABT point of view, there are few genotoxic compounds and there are and their modes of action and their mechanism I have listed in the table. So first compound is a cyclophosphamide or cyclophosphamide monohydrate. So current status is currently in use in the market, and the mode of action is the compound is a clastogenic. So what is the mechanism of genotoxicity? So DNA cross-linking after the metabolic activation. So if we are using cyclophosphamide monohydrate as a positive control, there should be metabolic activation system. Then the second is a mitomycin C, which is also currently in use, also anti-cancer. Then mode of action, it's clastogenic. Then mechanism of genotoxicity, it alkylates DNA, causing interstrand crosslinks. Then the third is a vincristine, which is also in use, and the mode of action is anugenic. So it disturbs the microtubule formation, and which leads to the chromosomal misaggregation. Then aflatoxin B1, which is naturally occurring compound. So mode of action is clastogenic, structural chromosomal aberrations, and mechanism of genotoxicity is it metabolized to epoxide, and it gives DNA ducts. Then next is the ethylene oxide. So it's industrial chemical. Again, mode of action is clastogenicity, and mechanism is direct alkylation of DNA bases. Then next is thalidomide, which is withdrawn. The mode of action is anogenic, which disturbs mitotic spindle formation and angiogenesis. Then diethyl still bestrol, which is also withdrawn, and mode of action is clastogenic. So mechanism of genotoxicity is oxidative stress, receptor mediated DNA damage. The next is a benzopyrene, which is an environmental pollutant. A mode of action is clastogenic. So mechanism is it forms bulky DNA adducts after metabolic activation. So if you are using benzopyrene as a clastogenic agent or as a positive control in the assay, it should be with metabolic activation system. Then graciofulvin currently in use, antifungal, and mode of action is anugenic. So mechanism is it interferes with mitotic spindle function. Then the last one is a chlorambucil, which is currently in use, which is also anti-cancer drug, and mode of action is it's clastogenic. So mechanism of action is it alkylates DNA, strand breaks and it crosslinks. So these are different in use currently in market, or a few genotoxic compounds. Those are withdrawn from the market, and there is mode of action and mechanism of genotoxicity. So this is all about genetic toxicology.